Welcome back. Today we're going to talk about Chapter 5, Microbial Metabolism. So we're going to look at both catabolic and anabolic reactions to generate ATP, but we're going to talk mostly about the catabolic metabolism. So if we look at microbial metabolism provides the ability to break things down, and then anabolic reactions gives us the ability to build things up. So we start out with complex molecules such as starches and proteins and lipids. Catabolic reactions break them down into simple molecules such as glucose, amino acids, glycerols, and fatty acids when we talk about our larger lipids. These simple building blocks then, through anabolic reactions, are going to be used to make complex molecules, and the recycling continues. In the process, catabolic reactions release ATP, whereas anabolic reactions use ATP to build things. The system, however, is not 100% efficient. So we see that there's a lot of energy lost in the form of heat, and unfortunately, that means that metabolism has to figure out ways to try to be as efficient as possible and capture as much energy as it can. So, in thinking about catabolic and anabolic reactions, metabolism is really pathways where you have a sequence of enzymes working together to produce a product. So you have starting molecule A, and then reaction 1 produces B, but all of these are going to be controlled by individual enzymes that work on that specific substrate. So that's why complex pathways, even knocking out a single enzyme, can have major effects on that pathways because they often are sequential. These enzymes are encoded by genes, and that's why if you have a mutation, even at the genomic level, this can have major implications in metabolism. We're not going to focus a lot on the enzymes themselves. We're going to look at the process of catabolism and anabolic reactions, but I do need you to remember that all of the pathways are going to have enzymes that are going to push those reactions forward. So in energy production, ATP is going to be the universal exchange, but we also have to harvest energy in an efficient way, and much of this involves what are referred to as oxidation-reduction reactions, or you may have heard them referred to as redox reactions. ATP is always going to be the universal energy exchange, and that's going to be for bacteria or eukaryotic cells. It is a molecule that is uniquely designed to be able to utilize in the transfer of energy. It has high energy unstable bonds that allow those phosphate groups to be removed. So even though this sounds like this should be a direct route, a lot of the generation and transfer of energy is not done directly. We'll see that energy production involves oxidation and reduction reactions in the generation of ATP. So I'm sure you've heard of oxidation reduction reactions already. And the simple definitions: oxidation, the addition of oxygen; reduction, the loss of oxygen, isn't going to be very useful for our discussions of metabolism. What's more important is looking at oxidation as the removal of electrons or hydrogen ions. Reduction is then the gain of electrons or hydrogens. In reality, we're going to start being more interested in where those hydrogens are going as a way to be able to follow the energy or potential energy throughout metabolism. So these redox reactions are always going to be coupled. In the figure here, we see A is going to donate its electrons to B. So in the process, A undergoes the process of oxidation to be oxidized, and B, in gaining the electrons, has undergone the process of reduction and is now going to be reduced. Biological reactions are often oxidations. So these are also referred to as dehydrogenations. So we can think of them as where are those hydrogens going, who are they getting passed off to? So in the example here, we have a compound A that has two hydrogens. So as AH2 becomes oxidized, those hydrogens are going to be passed off to a coenzyme, in this case NAD positive, and that NADH is now going to be reduced. So we're going to follow a lot of these dehydrogenations in looking at how we pass energy and eventually form ATP. In reality, when we talk about the food sources that cells take in, what we really want to do is utilize these oxidation reduction reactions to produce energy. So cells want to take in highly reduced compounds. Those are going to be things like sugars. The preferred sugar is always glucose. So sugars are carbohydrates. And so therefore, we see that they have many hydrogen atoms, and they then contain much potential energy. As we saw in the previous slide, those reactions have to be coupled. So if we have organic molecules that are going to have lots of hydrogens on them, they are obviously going to go through oxidation, and the organic molecules are going to become oxidized. So really, metabolism is just taking in these highly reduced compounds and trying to oxidize them over and over again. Well, since these reactions have to be coupled, that means that there's coenzymes such as NAD positive and NADP positive. These are commonly carrying the hydrogens in these redox reactions. We're going to focus on NAD positive. NAD is nicotinamide adenine dinucleotide, and the positive designates that this is the oxidized form. So it is going to be an electron carrier, an essential coenzyme that is going to be willing to accept those hydrogens. So as NAD positive accepts the hydrogens, it undergoes reduction, and then you have NADH, which is the reduced electron carrier. We're not going to talk a lot about NADP because that is involved in anabolic reactions. We're going to focus mostly on catabolism in this lecture. So dehydrogenations are very important. These are the biological oxidations, and NAD is going to be a vital coenzyme that's going to help us capture a lot of that energy. So NAD positive, the oxidized form, again, is going to be willing to accept those hydrogens. And now as the reduced form, NADH, it actually contains lots of potential energy. However, similar to foreign currency, unless we can get that NADH into a usable form, we can't really spend it. So the universal exchange is ATP. So a lot of what we're going to talk about is how we get this potential energy into a usable form in the form of ATP. So ATP obviously is important as a universal energy exchange molecule, and therefore we see that most of the time we're generating ATP from the precursor ADP. So there's two phosphates, a little bit of energy, and another unstable phosphate bond can be added. So we're going to look at the different types of phosphorylation, which means that we've added the phosphate group to ADP. There's three types of phosphorylation. The first type, substrate level phosphorylation, is when you transfer a high energy phosphate to ADP, but it comes directly from another molecule that it is attached to. So we see here a carbon backbone, and it has an unstable phosphate attached. It is going to be very willing to give up that phosphate if ADP is around. Then we end up with that organic molecule and produce some energy in the form of ATP. In this direct transfer, no oxygen or other inorganic electron acceptors are required, and therefore we can make these high energy ATPs from intermediates throughout metabolism. However, in reality, not much of our ATP throughout metabolism actually comes from substrate level phosphorylation. Where most of our energy is generated is from oxidative phosphorylation. In oxidative phosphorylation, electrons are removed from organic compounds, and then they're transferred in sequence to the electron transport chain. In that chain, they then pass the electrons to the final electron acceptor, which in aerobic cellular respiration is going to be oxygen. But bacteria, many things can be anaerobic as well, such as Clostridium, our gram-positive rod that we talked about when we looked at endospores. Those types of organisms carry out anaerobic cellular respiration and use things like nitrates and sulfates as the final electron acceptor. In part B of this chapter, we'll talk about fermentation of organic compounds and see how that low energy process is very beneficial in creating a lot of our dairy and alcoholic beverages. This chapter, or this part A of chapter 5, is going to focus on the catabolism, and a lot of the information is going to be about oxidative phosphorylation. So this part A will be a little bit long, part B is much shorter. So energy is going to be released in this process of oxidative phosphorylation and help us make the majority of our ATP, whether we're talking about bacterial cells or eukaryotic cells. Oxidative phosphorylation, it's kind of the big umbrella of the process, but this energy is actually going to be harvested by a process referred to as chemiosmosis. Osmosis is when we put something across the membrane, and in this case, the chemi part is going to refer to those hydrogens that we're going to be moving throughout metabolism. The process is also dependent on those essential coenzymes, so we'll see NAD positive having a major role, and there's also a flavin form of the adenine dinucleotide that will be involved as well. There's also photophosphorylation, but we're not going to have time to talk about the photosynthetic bacteria. So suffice it to say, light causes chlorophyll to give up its electrons. The energy is released from the transfer of electrons, oxidation of the chlorophyll through a system of carrier molecules unique to photosynthesis, but sharing the other catabolistic mechanisms that we'll see in oxidative phosphorylation to generate ATP. But we're going to talk about the processes that are going to be involved in substrate level and oxidative phosphorylation. Before we start talking truly about catabolism, just a little bit of information about enzymes and their action. When we talk about enzymes, enzymes are proteins, and when it's just the protein itself, we usually refer to that as the apoenzyme. Many of these proteins have essential cofactors or coenzymes. Cofactors are non-protein components, whereas coenzymes are organic cofactors. So we're going to focus on some important coenzymes, as I said before, NAD positive, FAD, and coenzyme A. The holoenzyme then is going to be the apoenzyme plus the cofactor or coenzyme as needed. When we look at metabolism, many of the reactions would actually still take place without enzymes, but very, very, very slowly. And so therefore, the reaction rate can be increased by enzymes, and that's why we think of all metabolism as being these sequences of enzymatic reactions. So in order to lower the reaction rate, enzymes are going to be what we think of as our metabolic processes. In the energy requirements, we see if we start with the substrate, here A and B are reactants within that substrate. The blue line indicates what would happen without enzymes. The activation energy without the enzyme is quite substantial, and it would take a long time before we got our end products of A and B. When you add enzyme, reaction with the enzyme lowers that activation energy, and therefore things are going to proceed much more quickly. So that's why when we talk about metabolism, we often talk about the enzymes that are actually driving metabolism forward. Enzymes can usually be recognized by their ASE ending. That usually is coupled with words that refer to what's happening in that reaction. So a transferase, for example, transfers functional groups. We're also going to look at the important coenzymes, as I said, NAD positive, FAD, and coenzyme A. Because we're not going to spend a lot of time on the anabolic building reactions, we're not going to talk about NADP, but it is an important coenzyme. So metabolic pathways really are the sequence of enzymatic reactions. And we see that one is going to drive the next one as the new product B is made, then another enzyme comes in and turns that into product C, and so forth until we get to our desired end products. So we're going to look at these pathways that are going to be involved in generating ATP in the bacterial cell. As I said, we're going to focus on carbohydrate catabolism. All cells prefer carbohydrates as their energy source, so we'll take a look at glycolysis, the Krebs cycle, and the electron transport chain and see how all of that fits together. So for most organisms, they are going to prefer glucose as their preferred carbohydrate. And as I said, most organisms simply are going to keep oxidizing those carbohydrates until they get as much energy as possible out of them. So the breakdown of carbohydrates to release energy is going to happen in several major pathways. Glycolysis will be the first, then Krebs, and then our cashing in our potential energy, taking it to the bank, so to speak, and the electron transport chain. We see here what glucose looks like as the most common carbohydrate. It's a 6-carbon carbohydrate, and it's basically going to be the start of catabolism. So in our overview, we're going to talk about respiration first. From glycolysis, that 6-carbon glucose molecule is going to be turned into pyruvic acid, and at that point there's a juncture to decide whether things go through respiration or fermentation. If you're talking about eukaryotic cells, this is driven by the fact of is oxygen present or not. If oxygen is present, then you're going to usually do respiration, and that is going to give you the most bang for your buck. If oxygen is not present, then cells will start to use fermentation. When they start to use fermentation, that's when you start to have buildup of the fermentation byproducts, acids such as lactic acids, and that's when your muscles start to feel tired as you've used up a lot of your energy. Bacterial cells are only single cell, so we're going to see things in more microbial diversity of there's going to be cells that are going to only be able to carry out aerobic respiration. Some, like Clostridium, that can only carry out anaerobic respiration. Some that can only do fermentation. But we do, similar to our own cells, have some switch hitters that can go back and forth between respiration and fermentation, depending on the conditions. But for now, we're going to focus on respiration and see how we generate our potential energy and then cash that in the electron transport chain. To get started, we're going to talk about glycolysis. Glycolysis is the oxidation of glucose to pyruvic acid. Glucose is a 6-carbon molecule. Pyruvic acid is a 3-carbon molecule. So therefore, we're going to be able to generate 2 pyruvic acids for every 1 molecule of glucose. In the process, we're going to generate a little bit of direct energy in the form of ATP and a little bit of potential energy in the form of NADH. So again, if we start to think of NADH as this foreign currency, we have the ability to have some money for exchange, but we have to get it into the right form to be able to actually use it. So in glycolysis, we actually have to spend or put in some ATP to get things started. That is referred to as the preparatory stage. And we see in the preparatory stage that glucose is first going to be phosphorylated, and we end up making what's referred to as glucose 6-phosphate. You don't see this happening, but rearrange some hydrogens and you end up with fructose 6-phosphate. We put in another ATP, and this makes a nice symmetrical molecule, what is referred to as fructose 1,6-diphosphate, and it has the phosphates on both the 1 and the 6 carbon. In these processes, I don't want you to know every single intermediate. I will point out the ones that you know, but I want you to follow how many carbons, so the blue balls, and I want you to also remember where are the phosphates going? What are we transferring where? So in this reaction, once we have the fructose 1,6-diphosphate, this gets cleaved into two important intermediates that you do need to remember. The DHAP, which is dihydroxyacetone phosphate, and the GP is glycerolaldehyde 3-phosphate. This is a forward and reversible reaction between the two. However, DHAP is readily converted to GP. GP is the only thing that gets further oxidized in metabolism. The way to think about this is think of the rest of metabolism as like a funnel waiting there for the glyceraldehyde 3-phosphate to be made. So as soon as it's made, it drops down the funnel and goes through the rest of metabolism. And that way we see that the push is always going to be from the dihydroxyacetone phosphate to making GP. Once we've made our glyceraldehyde 3-phosphate, then we can go through the energy conserving stage. Steps 6 to 10 are going to now finally allow us to make some ATP. Please note over to the side in the metabolic reaction pathway, everything is multiplied by 2. Because the dihydroxyacetone phosphate was converted into a second molecule of GP, both of those GP molecules are now going into the energy conserving stage, so that's why everything is going to be multiplied by 2. So the first thing that happens, we generate a little bit of potential energy in the form of NADH. We also see an input of inorganic phosphate that is going to make another unstable phosphate bond, giving us 1,3-diphosphoglyceric acid. This is a very unstable bond, and so we produce rather quickly 2 ATP. This leaves us then with the 3-phosphoglyceric acid, a little bit of rearrangement, and we end up with that phosphate on the 2 position, kick out some water, and that makes a very unstable bond, and the phosphate is easily removed from what is referred to as phosphoenolpyruvic acid. PEP is kind of what puts the pep in our step, and we see that another 2 ATP are going to be generated as we make our final product of glycolysis, the pyruvic acid. So again, we get 2 pyruvic acids. These are 3 carbon molecules, and in the process of this energy conserving stage, we've made 4 ATP and 2 NADHs in the form of potential energy. However, we have to think about our bookkeeping. We had to spend 2 ATP to get things started. So when we look at this and we've generated our 2 pyruvic acid, our 4 ATP, and our 2 NADHs, we only have a net gain of 2 ATP because we have to subtract out the 2 ATP that we initially put in. But we still have 2 ATP as our net gain, and we also have our 2 NADH molecules as potential energy. Glycolysis is the oldest metabolic pathway. Glycolysis was the original pathway that allowed cells, ancient cells, to be able to start utilizing carbohydrates. If something is only growing by glycolysis, at maximum, it can only make 2 ATP. So those types of cells are going to grow very slowly. Therefore, it was important metabolically that cells figured out how to get more energy out of glucose, more bang for their buck, but glycolysis, being the most ancient, is kind of the building block of generating that pyruvic acid, and then other metabolic pathways went off from there, things like the Krebs cycle. So here we see a detailed outline of the reactions of glycolysis. Again, recognize where the ATP is being put in and where it is being produced. Our two important intermediates, the DHAP and the GP, are glyceraldehyde 3-phosphate. After the gray line there, we start to see that we actually generate some ATP. 4 ATP, but our bookkeeping means we have to subtract the 2 that we originally put in, so a net gain of only 2. And again, potential energy in the form of those 2 NADH will become important later as we're trying to make ATP. If something is important in biology, usually there's alternatives. So it makes sense that there's alternatives to glycolysis. We have the pentose phosphate pathway and the enterodeuterose pathway as well. For the pentose phosphate pathway, this may also be referred to as the hexose monophosphate shunt. It uses pentose's 5 carbon molecules and NADPH, the reduced form of the NADP. This operates with glycolysis and is important in making or synthesizing nucleic acids. So nucleic acids are needed to make DNA, RNA, so obviously this is a very important shunt in order to make our DNA and RNA that has to operate with glycolysis, as we're using glycolysis to get energy. It's also true that certain amino acids come from this pathway as well. The enterodeuterose pathway, this does not involve glycolysis, and it's just another way to oxidize glucose to pyruvic acid. It produces NADPH and ATP, but it has limited application. It's generally found in gram-negative bacteria, things like Pseudomonas, and some of our soil organisms, things like Rhizobium and Agrobacterium. Important to those organisms, but we're going to focus mostly on glycolysis and where we go from here. So, as I said, we're going to have this important decision of we've made pyruvic acid, and now what do we do? So for the rest of this discussion, we're going to go down the left-hand pathway and talk about respiration. In part B, we'll talk about fermentation. So cellular respiration is going to generate ATP. Molecules are going to be oxidized. Again, most of metabolism is just taking in something highly reduced and just keep oxidizing it and oxidizing it. So the oxidation of molecules is going to help liberate the electrons for the electron transport chain. In cellular respiration, the final electron acceptor is an inorganic molecule. As we said, aerobic respiration, it's going to be oxygen. Anaerobic respiration, other inorganic molecules, as we'll see, things like nitrates and sulfates come to mind. An essential feature, the electron transport chain. That is really going to be the bank where we're going to cash in our potential energy and exchange it into ATP. The big umbrella process is that ATP is going to be generated by oxidative phosphorylation. We're using oxidation to be able to eventually make ATP by phosphorylation. We're going to get ready to go into Krebs, but before we can go into the Krebs cycle, we have this very important intermediate step. Pyruvic acid from glycolysis is going to be oxidized and also decarboxylated. So we're going to kick off some CO2 and turn pyruvic acid into a 2-carbon molecule of acetyl-CoA. It's referred to as acetyl-CoA because as well we have a coenzyme that is going to contribute to making this very small acetyl-CoA molecule. There's also some potential energy made. We have made NADH. Remember, since we generated 2 pyruvic acids, we're also making 2 acetyl-CoAs, and we're also generating here 2 NADH molecules. So that acetyl-CoA is going to be what gets the Krebs cycle to turn and start to make some more potential energy and a little bit of ATP by direct substrate phosphorylation. The Krebs cycle, it looks big and scary, but it's really just a recycling of a 4-carbon molecule. We start out with oxaloacetic acid, and then in reaction 1, it is going to be converted into citric acid as the acetyl-CoA is added. That's why sometimes you'll hear the Krebs cycle be referred to as the citric acid cycle. The citric acid is then going to have some rearrangements of hydrogens, and that's going to make isosateric acid. Then in step 3, we see the first of our potential energy in the form of NADH being produced, and we kick off a little bit of CO2. This gets us down to a 5-carbon molecule, alpha ketoglutaric acid. We then basically do the same thing. We make a little bit of potential energy in the form of NADH, kick off another CO2 molecule to get a 4-carbon molecule, and we add in the coenzyme CoA. This produces a 4-carbon succinyl-CoA, and that CoA is on there by a very unstable bond, and that is going to allow us to make a little bit of ATP by substrate or direct phosphorylation. So succinyl-CoA to succinic acid is an important reaction to remember, because that is going to be the only place in the Krebs cycle where we get ATP by that direct or substrate level phosphorylation. Succinic acid to fumaric acid in step 6 is also an important reaction to remember, because this is the only place in metabolism where we are using the flavin form of the adenine dinucleotide, and we're generating FADH2 here. Fumaric acid then is converted to malic acid, and then our last step, we get back to where we started. Malic acid is converted into the oxaloacetic acid, and we produce another NADH. Again, because we put in 2 acetyl-CoA, we can make the Krebs cycle go around twice. So everything we see in the cycle gets multiplied by 2. So if we look at our bookkeeping, we have 4 CO2s, 6 potential energy in the form of NADH, 2 potential energy in the form of FADH2, and 2 ATP. So the Krebs cycle, again, recycling of the 4-carbon oxaloacetic acid. First thing is acetyl-CoA comes in to make citric acid. We generate potential energy in the form of NADH and FADH2, and a little bit of ATP by direct substrate level phosphorylation as succinyl-CoA goes to succinic acid. The big deal about the Krebs cycle really, though, is the potential energy. At the end of the electron transport chain, a bacterial cell is going to be able to generate 38 ATP. So right now, we've only generated 4 ATP through glycolysis and the Krebs cycle. The big payoff is going to be taking that potential energy that's been produced in the Krebs cycle and a little bit in glycolysis and the intermediate step, and now harvesting that or taking it to the that is going to happen in the electron transport chain. So the electron transport chain is going to be the place where we play past the electrons. The electron transport chain has a series of carrier molecules that are going to become oxidized and reduced as electrons are passed down the chain. And then it sets up a gradient that is going to allow us to carry out the process of chemiosmosis. So we're going to see these protons are going to be generated that want to get back across the membrane. So let's take a look how that actually happens. First of all, just a reminder, the electron transport chain for a prokaryote is going to be in the cell or plasma membrane. That is going to be analogous to the inner mitochondrial membrane of a eukaryotic cell. So for a eukaryotic cell, the electron transport chain is in the mitochondria in that inner mitochondrial membrane that's analogous to the cell membrane of prokaryotes. And if you remember what we talked about with the endosymbiotic theory, all those mitochondria were at one time those ancient prokaryotes. It makes sense that they became the powerhouse of the cell because now eukaryotes have lots of mitochondria, especially in cells like muscle cells that need to be very active. So the electron transport chain is going to harvest the potential energy that is found in NADH and FADH2. So we're going to utilize both the nicotinamide adenine dinucleotide and the flavin adenine dinucleotides that we've previously generated, but the useful form is the reduced form, NADH and FADH2. So carbohydrate metabolism, our last process, is going to be looking at what happens at the electron transport chain. My analogy of NADH and FADH2 being potential energy means that the electron transport chain is basically going to act like the bank. The bank is going to be where we take that foreign currency and cash it in and turn it into something useful in the form of ATP. The electron transport chain is made up of three classes of carrier molecules. You have flavoproteins. The first one in the chain there is the FMN, which is flavomononucleotide. The flavin is a coenzyme that is oxidized and reduced. We also have ubiquinones. In the chain, we see coenzyme Q, which is a non-protein carrier. And then the rest of the chain is made up of a variety of cytochromes. Cytochromes use iron that is oxidized and reduced. That is why when you don't get enough iron, you don't produce enough energy. You feel tired and sluggish. Well, even though we don't have bacteria that feel tired and sluggish, it's a true statement that without iron, bacteria are not going to be able to grow and metabolize. They won't be able to divide and increase in population. So these carriers are going to accept and release electrons or protons as well. So we're going to play the game of pass the electrons, but in that game, we're also going to generate these protons. A lot of people like to think of the electron transport chain as this energy hill. So they like to think of the flow of electrons going downhill, starting from the high energy top of the hill at the FMN, which is the first member of the electron transport chain. And NADH is going to bring us the most energy because it feeds in at the top of the hill. FADH2 comes in at a lower energy level, at the level of coenzyme Q, but all of these are going to feed into the various cytochromes until we keep passing the electron down to the final electron acceptor, which in aerobic respiration is going to be oxygen with a byproduct of water. So the purpose of the bank here is to cash in our potential energy, our currency, but it happens that the exchange rate at the bank is very good. So NADH and FDH2 are oxidized and the electron carriers are going to then contribute to the production of ATP. The exchange rate is going to be for every 1 NADH, we're going to be able to generate 3 ATP. For every 1 FADH2, we're going to be able to generate only 2 ATP. So how does it actually happen? Playing past the electrons to the final electron acceptor, it moves things, but it doesn't really explain how we're actually getting that ATP. The secret is that some of the members of the electron transport chain also are going to be proton pumps. So FMN, coenzyme Q, and cytochrome A3 are also going to release protons. Those hydrogens, those protons, are going to want to come back across the membrane, and as they do so by chemiosmosis, we're going to harvest the energy and turn it into ATP. So here's what the chain actually looks like. It's embedded in a membrane. Again, for a bacteria, this is going to be at the cell membrane. For a eukaryotic cell, this is going to be the inner mitochondrial membrane within the mitochondria. But we're going to focus right now on what happens with NADH. So in step 1, we see NADH is going to come into the first member of the electron transport chain, which also happens to be the first proton pump. So as NADH comes in and is going to be oxidized to NAD positive, we're going to see the flavin mononucleotide is now going to act as a proton pump to put 2 protons outside of that membrane and also pass the electrons to activate the rest of the electron transport chain. It then goes to the second pump. Ignore for the moment the FADH. We're going to talk about that story in a moment. So the second pump gets activated and we pump out 2 more protons across the membrane, pass the electrons, and the cytochromes now get activated. And then we finally get to the third proton pump, cytochrome A3. Another 2 protons are then pumped out. So each proton pump, FMN, coenzyme Q, cytochrome A3, each of those pumps out 2 protons. So that's going to give us a total of 6 protons to the outside of the membrane. Those protons now want to get back across the membrane, but the only place that they can do that is by a specialized channel referred to as ATP synthase. As the name implies, this is going to be an enzymatic channel that allows us to generate ATP. As 2 protons pass through the channel, you get 1 ATP. So therefore, when NADH comes into the electron transport chain, it generates 6 protons. Those 6 protons coming across the channel 2 at a time allow the generation of 3 ATP. So that's why we say our exchange is going to be for every 1 NADH that comes in, we generate 3 ATP. Now, if we look again at the FADH2 that comes in at a lower energy level, it's bypassing the first pump, so it doesn't make those first 2 protons. So when FADH2 comes into coenzyme Q, it's going to activate that pump, put out a pair of protons, pass the electrons to all the cytochromes, and then the last proton pump, cytochrome A3, again is going to put out 2 protons. So for FADH2, it can only generate 4 protons. Therefore, it can only generate 2 ATP. So that's why the exchange for every 1 FADH2, we can only generate 2 ATP because we're only making 4 protons. So the ATP synthase channel is very important. By having these proton pumps, we're creating that concentration gradient. We're going to have a high concentration of those protons to the outside of the membrane. They want to get back across. That's the process of chemiosmosis, but we're only going to allow them to come back through the ATP synthase channel. So they're going down their concentration gradient from high to low, but they have to use the facilitated protein, the ATP synthase channel. So every time 2 protons come across, we make 1 ATP. So that's why chemiosmosis as a process is very important. Those 3 proton pumps are going to build up the protons on the outside of the membrane, and similar to water behind a dam, that's stored energy. That accumulated proton motive force is now going to want to come back across the membrane, and if we only allow it to come through the special channel, the ATP synthase, we're now able to synthesize ATP. So that's how we take the potential energy that NADH, that FADH2, and actually harvest it to make ATP. Respiration, as we said, can be aerobic or anaerobic. So the biggest bang for our buck is going to be when a cell carries out aerobic cellular respiration. it's going to be able to make 38 ATP for every 1 glucose. Anaerobic respiration, the final electron acceptor, is not oxygen, but still inorganic. Suffice it to say, because we're not going to have enough time to really talk about all the intricate detail of anaerobic respiration, but anaerobic respiration yields less energy than aerobic respiration because only part of the Krebs cycle can be utilized. The electron acceptors in anaerobic respiration are things like nitrates and sulfates and carbonates. So one of the products is hydrogen sulfide for some organisms, which literally smells like rotten eggs. And so we often associate these anaerobic organisms, such as Clostridium, with some of these very interesting end products. So where does metabolism occur? For a prokaryote, it does not have membrane-bound organelles, it doesn't have mitochondria, so glycolysis, the intermediate step of taking pyruvic acid to acetyl-CoA, and the Krebs cycle all have to occur in the cytoplasm. The electron transport chain is in the cell or plasma membrane. For a eukaryote, glycolysis and the intermediate step occurs in the cytoplasm, but the acetyl-CoA needs to be shunted into the mitochondria because the Krebs cycle occurs in the mitochondrial matrix. So that's why it makes sense that the electron transport chain is at that inner mitochondrial membrane. So if we look at our bookkeeping, this table represents how much ATP, NADH, and FADH2 we've produced. So we've produced a total of 4 ATP by direct substrate level phosphorylation, 2 in glycolysis and 2 in the Krebs cycle as we went from succinyl-CoA to succinic acid. The big payoff is going to come via oxidative phosphorylation using the NADH and FADH2 that have been produced, but mostly NADH. So glycolysis gave us 2 NADH, the intermediate step, another 2, but Krebs gave us 6 NADH. So that's a total of 10 NADH molecules. FADH2 is only produced as we go from succinic acid to fumaric acid in the Krebs cycle. So we get 2 FADH2, but that's the only place in the Krebs cycle. So that's kind of what's produced. But what does that give us in the form of energy in ATP? So if we look at the ATP produced, we actually now have to take into consideration our exchange of our currency. So the ATPs that we talked about on the previous slide doesn't change. That was just simply by direct or substrate level phosphorylation, 2 from glycolysis, 2 from Krebs, so we got 4 there. But the big payoff is going to be by oxidative phosphorylation via the exchange in the electron transport chain. So if every 1 NADH gives us 3 ATP, that means that all the NADHs that we've generated, we're going to multiply that by a factor of 3. So if we had 2 of the NADH in glycolysis, multiply that by 3, that gives us a total of 6 ATP, another 6 ATP in the intermediate step, and the big payoff, 6 NADH molecules are produced from the Krebs cycle. As those go through the electron transport chain, that 1 NADH equaling 3 ATP means we multiply by 3, and that gives us a big increase in 18 ATP. So if we add those up, we actually get 30 ATP from NADH by oxidative phosphorylation. The FADH2, this gives us some, but not as much. The exchange rate was 1 FADH2 equals 2 ATP. So in the Krebs cycle, as we went from succinic acid to fumaric acid, we generated 2 FADH2, but the exchange rate is only going to be 2 because 1 FADH2 equals 2 ATP, so we multiply that 2 FADH2 by our 2 exchange rate, and that gives us 4 ATP. So in that way, we see we have a total of 38 ATPs produced in prokaryotes. So let's play a little game with looking at how we're producing this energy. If we knock out the enzyme so that DHAP, our dihydroxyacetone phosphate, can't be converted to GP, our glyceraldehyde 3-phosphate, how many ATP could this mutant cell produce? If you think about it, the step here is right before we go into the energy producing. So we're at the very end of the preparatory stage, and if now we can't convert that DHAP into our GP, that means we're only going to have one GP going through the rest of the process. So a good guesstimate would be, all right, probably about half the amount of energy. So if we're supposed to generate 38 ATP, all right, probably about 19 ATP. That's a good guess, but the answer is actually 18 ATP. If you do the bookkeeping, we see that above the gray line to the right-hand side, if we only have one molecule of glyceraldehyde 3-phosphate being put into our making ATP in the next stage, we no longer have the 2 in front of all of our intermediates. So it would only be one going through the energy producing phase. And so in that way, we would only be able to produce 2 ATP. And therefore, if we had to spend 2 to get it started in glycolysis, we actually don't end up with any ATP in glycolysis. We would still have the one NADH that was produced after the gray line there as we went into our energy producing stage. But again, we're not going to be able to multiply by 2 since the DHAP didn't get converted to a GP to be able to continue through the intermediate step in Krebs. So everything else. If we look at another theoretical mutant, this will help us to understand what's happening at the electron transport chain, thinking about our proton pumps. What if we mutated the cytochrome A3 pump so that it could no longer pump those protons across the membrane, but could still pass the electrons? How would that change the amount of ATP produced? Well, if we look at our diagram again, and we're knocking out the third pump, so that's cytochrome A3, let's focus on the exchange for the NADH right now in this mutant. So NADH is going to come into the first pump as usual, the FMN, makes our 2 protons there, passes the electrons to coenzyme Q. We get another 2 protons. Now we pass the electrons, but we get to the third pump, the cytochrome A3. It can still pass the electrons and pass those off to the final electron acceptor, but it can't pump. So what's going to happen is that this is only going to be able to generate 4 protons. Those 4 protons are still going to go across the ATP synthase, but remember, for every pair, we get 1 ATP. So if we only have 4 protons now being produced in this mutant, the energy exchange is only going to be 2 ATP for this mutant when we have 1 NADH coming across. If we think about this, not pictured here, but if we think about the FADH2, it bypasses that first pump, so we don't get those first 2 protons anyway. Now the FADH2 is only using the coenzyme Q pump and is only going to make 2 protons, and so that is simply only going to be able to come across and give us 1 ATP. So if we tally up our score, so to speak, for our ATP, we see that this exchange is going to take down the amount of ATP by affecting our exchange rate. So we're not going to do anything to the substrate level phosphorylation. We still have 4 ATP there. However, in oxidative phosphorylation, it's going to change the exchange rate, both for the NADH and the FADH2. So if we knocked out that third pump, then when we look at the NADH, instead of being able to multiply by 3, we would only be able to multiply by 2, because we're now only going to be able to generate 2 ATP for every 1 NADH that goes through. So therefore, if we tally that up, that would mean that if we only have 2 out of 3 pumps active, we only get 2/3 of the amount of our ATP from NADH, so only 20 ATP. For the FADH2, the exchange rate normally is 1 FADH2, and that gives us 2 ATP because normally we utilize the 2 pumps, the coenzyme Q and the cytochrome A3, but if we knock out the cytochrome A3, all we have are the 2 protons from that coenzyme Q pump. So that means that the exchange rate really is just 1, and so we're only going to get 2 ATP from the FADH2 that goes through the electron transport chain in this mutant. So for this theoretical mutant, it would only be able to generate 26 ATP, and obviously this might not be a viable mutation, but it's a fun way to think about the electron transport chain. Note for aerobic respiration in eukaryotes, there's a little bit of diversity in the bookkeeping. Aerobic respiration in eukaryotics are only going to produce 32 to 36 ATP. So that's going to be less than the 38 ATP produced from 1 molecule of glucose that we saw for a bacterial cell. So that seems to not make sense, but some energy is lost when electrons are shuttled across the mitochondrial membrane to be able to utilize that in the electron transport chain. Remember, glycolysis is in the cytoplasm, and so we have to get things, this is acetyl-CoA and everything into the mitochondria. So what's happened in a eukaryote is that it's not getting as much for every single molecule of glucose because it's putting it into the mitochondria to be exchanged at the electron transport chain. However, it's made up the difference because a eukaryotic cell has lots of mitochondria, the powerhouse of the cell, so it doesn't matter that each mitochondria is doing a little bit less energy. So in that way, it allowed eukaryotic cells to become multicellular for the fact that you started to be able to generate lots of energy because of having multiple mitochondria per cell. So that's the summary of aerobic respiration. We started off with glycolysis, a 6-carbon glucose molecule, converted that to 2 3-carbon pyruvic acid molecules, generated a little bit of ATP, 2 ATP in fact, and a little bit of potential energy in the 2 NADH. We then had the intermediate step to turn that pyruvic acid into acetyl-CoA, kicked off from CO2, and got a little bit more 2 NADH in the process. Big payoff in potential energy comes from the Krebs cycle. 6 NADH and 2 of the FADH2 means that in total, we're going to have 10 NADH and 2 FADH2 molecules, taking those to the bank in the electron transport chain to be able to produce 34 ATP. The other place where we generated 2 ATP as we went from succinyl-CoA to succinic acid in the Krebs cycle by substrate level phosphorylation. We now have accounted for all 38 of our ATP from 1 molecule of glucose in a prokaryotic cell. So that's the overview. As I said, next time we're going to, in part B, which is much shorter, talk about fermentation and all the good things we get from fermentation. As always, make sure you check your understanding what catabolism and anabolic reactions look like, what you need to remember about glycolysis, the Krebs cycle, and generating ATP through the electron transport chain. And we'll pick up there in part B in talking about our fermentation and the process that gives us a low energy yield.